That's great. So welcome everybody to the Marine Biology live session. This is the fifth in the series. It's a real pleasure and an international reach to invite Sonia Batten to join us for her morning, all the way from Vancouver. Sonia is the Executive Secretary of PICES, and prior to that worked as a biological recorder for the Continuous Plankton Recorder, CPR, and Sonia was the director of the North Pacific CPR, so she's well known to many people here at the MBA. She's going to talk to us this morning about a project with NASA on the Interdisciplinary Science Programme about life in the moving ocean and a collaboration between the MBA and North American scientists working on marine ecosystems and the Northeast Pacific. So yeah, it's a great pleasure to introduce Sonia. I'll stop sharing my screen now, and hopefully Sonia should appear. Good morning, Sonia.
Good morning, John. Thank you very much.
So yeah, if you're able to share your screen now.
Hopefully you can see that. Let me know if that's working.
That's working fine. Thank you.
Perfect. Thank you. Good afternoon, everybody in the UK. It's nice to be talking to you from Vancouver Island. So East Meets West is that link between this MBA project and colleagues out here in the Northwest Pacific. It's a very collaborative project. And in fact, I'm probably the person who's done the least amount of work on this project, but I'm the one who gets to talk to you. So I will do my best to describe the work that we've been doing. So John mentioned that this is a NASA-funded study. So NASA has an interdisciplinary science program, Life in a Moving Ocean. Everyone knows that NASA is involved in space research and looking at the ocean using satellites. So the goal of our study was to better understand relationships between open ocean dynamics and ecosystems at medium to large scales. And I put this image up here of the Earth from space, which is focused on the Pacific Ocean. And you can see that it takes up about almost half of the globe, and that's ocean, and it's incredibly difficult to understand what's going on in that ocean, or it's also very expensive. And one advantage of satellites is that they cover a very large area of ocean. And one advantage of the other program I'm talking about today, the Continuous Plankton Recorder, is it also covers very large areas of the ocean. So it was a natural coming together of these two ways of looking at what's going in the ocean. So this project uses data from satellites. I'll go through a little bit of what we've been focusing on. Time series of plankton, which is the bottom of the marine food chain. We collect that using commercial ships. And we also have a database of seabird observations that we've been using. And the seabird observations are made by a huge variety of researchers over the years that have been gathered together in one place. So satellites can see quite a few of the surface oceans properties, and I've got three that I'm going to focus on. So the first one here is sea surface temperature, and over on the left there's an image of sea surface temperature anomaly, so difference from the average sea surface temperature that happened in a fairly recent marine heat wave in the Northeast Pacific. So temperatures were around 3 degrees warmer than average, which is a lot for marine organisms to deal with. This event was also called the blob because it shows up in these satellite images as a blob of warm water. And then over on the right-hand side of the screen, I've got sea surface temperature from off of the east coast of North America, showing the warm water coming up from the tropics and the Gulf of Mexico that gets deflected across and forms the Gulf Stream that keeps everybody in the southwest of the UK nice and balmy in the winter. But you can see, I think, from particularly the image on the right, that there is a lot of structure in the surface ocean that isn't necessarily visible unless you're looking at some of these properties that the satellites record. Then we have ocean color that you can see from space, and on the left this is the kind of true color of the ocean. And again, you can see patterns caused by organisms in the water, suspended sediment perhaps. There's clouds there too. And over on the right, if you recolor the information coming to the satellite and capture the wavelengths that are being reflected by plants and phytoplankton that live in the water. You can pick up areas where those are more dense. So this is an image of South Australia, that's Tasmania, and you can see that the red colours around Tasmania reflect higher densities of plankton in the water. And again, these ocean colour images show up the structure in the ocean at these larger scales, that it's not a homogeneous mixture. And final property that we can see is sea surface height. These are again sea surface height anomalies. This is a global picture, and the maximum anomaly is about 25 centimeters above and below the average. And again, the processes that are going on in the surface ocean, currents, wind-driven tides, or so on, can affect sea surface height, and these show up as positive anomalies in red and negative anomalies in green. So it's not really a huge difference, but it does again reveal structure within the surface ocean. So that's what the satellite data are showing us. And then I know many of you who are on this talk live today know an awful lot about the Continuous Plankton Recorder survey, but for the benefit particularly of people who'll be watching this later, or for our guests, just a little bit about the Continuous Plankton Recorder survey. So this is looking at plankton that form the bottom of the marine food chain. And now we're going below the surface of the ocean and looking at the organisms that are living in the surface waters. This is a program that's operating from the MBA. And most of its history has been based in the North Atlantic, but about 20 years ago, just over 20 years ago now, we started looking in the North Pacific, and that was my job for a number of years, was to manage that survey. So the survey uses commercial ships, so ships that are going from ports all around the world on regular routes, and they tow the instrument behind for us on those routes, and it collects plankton from the water. It's very robust. It can operate at very high speeds, which these ships typically go at. It's very cost effective because these are volunteer ships and they don't charge us for their time to go out and collect the samples. And the most important thing when you're running a monitoring program is that it's consistent over large scales. So our data set's consistent. We have the ability to sample at ocean basin scales, which is a very useful feature. And a little bit of detail. So this is one of the ships we use in the North Pacific. It's a container ship that goes from Seattle up to Anchorage. When they leave port, when they're clear of all port traffic, they deploy the plankton recorder for us behind the ship. And that sits in the water behind the ship, and basically it filters the plankton from the seawater onto a continuous length of filtering mesh. The way that it moves through the ocean, the propeller at the back controls the speed. So the mesh moves through the machine at a proportional rate to the speed of the ship. So we can easily reconstruct the voyage afterwards. And that image on the bottom right shows the length of mesh when it's come back into the lab. We end up with about 10 centimeters of mesh representing 18 kilometers or 10 nautical miles of the ship's voyage. And we cut those samples, that mesh into individual samples, and then analysts sit down at the microscope and look at the different plankton organisms, count them and identify them, and we build up a database of where the ship was, where the plankton came from. So that's what we have for the plankton information. And in the North Pacific, we started sampling in 2000. So this map here shows all of the different sampling locations. They're not all the samples on there. There's, I think, every other one. But in the first 19 years, we collected about 30,000 samples in total. We processed about 10,000 of them. All of them are in the archive, and we can go back and do that. And in fact, for this project, we did go back and reanalyze quite a few to give us some extra information in key areas. This is an ongoing project. So this is where we are at the moment, but it is ongoing. And today I'm going to be mostly talking about that northeast part, so the area that's bounded by Alaska to the north and Canada to the east. We call it the Gulf of Alaska, the Northeast Pacific. Those are sort of interchangeable terms. What I've got here is a movie. Hopefully it will work. I am not going to show all of it because it goes for the whole time series, but I want to give you the idea of how the ocean is dynamic. So this is sea surface temperature, and every now and again you'll see a black line, and that indicates one of our ships. So the Plankton Recorder survey is taking a snapshot of the ocean as those things go, as they go through. But the ocean is continually changing and moving, and the plankton that are in that ocean are being affected all the time. But in this, you can see the seasonal cycle of temperature. So this is a similar latitude to the UK. So we have very seasonal conditions. It starts off fairly cool, and then it warms up through the summer, and then cools down again in the autumn. And you can see that that movement of temperature isn't necessarily straight just south to north, but the currents that are going around the rim of the ocean are directing and deflecting that water, so that the areas around the coast are actually warmer than the open ocean. So that's sea surface temperature, and then from satellite information we can calculate the currents. So this is a modelling work done using satellite information to look at the currents. And one of the things that, again, you'll see the ship tracks from our sampling that cross there every now and again. One of the things you can see here is the very strong Alaska coastal current that starts life in the southeast of that image, moves along the coast towards the north, and then is deflected by Alaska. And you see these very strong currents. The colours are in meters per second, so those strong red ones indicate the fastest currents that basically then track along the coast of Alaska and out into the Bering Sea. And you can also see, again, the features, and I'm going to come back to some of these features, these eddies, which look like little rings, donuts, dots that are moving around in the current. So they're the reason for showing you these videos is that most of whenever I put up an image or the data, it looks like a snapshot, looks like a piece frozen in time. But just to make the point to you that the ocean is very dynamic and it's continually moving, and organisms living in it are continually experiencing change in their environment. Okay, so the plankton data that we collect consists of many hundreds of different taxa, different species or groups of organisms. And the term plankton includes a whole range of organisms, those that are teeny tiny from a few microns up to several centimeters in size, or technically up to quite large jellyfish. And it's one name, plankton, but it covers a huge array of different types of organisms, from those that are, photosynthetic and get all of their energy from the sun, to those that feed on each other, those that both photosynthesize and eat each other, carnivorous plankton. So there's very complex interactions within the plankton. Anyone who's worked with plankton knows that they're impossible to sample completely. No one instrument can cover the entire range of plankton organisms. So like all plankton samplers, some of them are well sampled by the CPR, some of them not so well. So I put some little smiley or frowny faces here just to kind of indicate that the ones that we do sample quite well, larger hard-shelled phytoplankton, small single cells, not so well. Crustacean plankton, so those are the copepods and krill, are well sampled, but the gelatinous plankton jellyfish, not very well sampled at all. But the point I want to make here is that we have a huge number of different species, and we want to simplify this when we're talking about how these organisms are important for fish, marine birds and mammals that live in the ocean. We have to try and simplify this, which is a real challenge. So the first part of our project that I'm going to talk about, which is close to being finalized now, is looking at the plankton communities across the Northeast Pacific and trying to come up with measures of that community structure. So for this, we've just focused on the zooplankton. And what I've got here is a sort of a summary plot of the community structure. So we merge the data into bins across the region, so by latitude and longitude, into rectangles. And then we compared the community structure that we found over the whole course of the time series. So this is an average pattern. And what we found was very consistent parts of the ocean for community structure. So the community structure is quite consistent in these regions, and each of these regions is different enough from each other to show up. So you've got, for example, one cluster that seems quite unique there near the Aleutian Islands in Alaska. That's cluster 7, and that was always significantly different from other clusters. But then you've got whole regions sort of towards the rim, that's clusters 1 and 2, that weren't that different from each other. So that's around the outer part of the gyre. And then you have some that, so cluster 6, which tends to be on the shelf, some coastal influence, and not much difference between the east and western sides of the Gulf of Alaska. And then you've got these other regions, clusters 3 and 4, which were in the center of the gyre and in the center of the Bering Sea. So those were more similar to each other than they were to the regions closer to them. And we found that those were very persistent over time. But one of the things that we had during our time series is actually quite a range in temperatures in the Northeast Pacific. So the top right graph there shows time along the bottom and sea surface temperature anomalies, so the difference from average. And you can see that in the middle part of our sampling, so the mid-2000s up to about 2012, we actually had colder than average years. But at the early part of the 2000s and in the later years, where we actually had a marine heat wave, we had some very warm years. So that gave us an opportunity to compare these communities between warm and cold years. And so on the bottom, we've picked the 5 warmest years and the 5 coldest years and looked at the pattern of sea surface temperature change. And you can see that the biggest difference in those changes is in that northern part of the region. So right up to the southern coast of Alaska is the areas that we're seeing the biggest changes between the warmest years and the coldest years. And if we looked at the plankton, the zooplankton communities, again, this is a similar plot to the one before showing the different clusters, but now we overlaid some color to show how those responded to changes in warm and cold years. So those that are shaded in red showed generally an increase in plankton numbers in warm. Those in blue showed a general increased numbers in cold years. The yellow areas down to the bottom right there, variable communities, they were sometimes higher, sometimes lower, but no clear response to temperature. And the grey areas in the center or the rim of the gyre there were sort of consistent regardless of temperature. So they didn't change very much at all. And whatever the temperature, they didn't change in a particular direction. So the important thing here is that although that northern region shows the biggest changes in temperature, very warm years and the very cold years, the zooplankton communities are actually very consistent in that region. But there are regions across this Northeast Pacific that show different responses to temperature. And this is the kind of information that's very useful for people who are looking at marine resources, so whether that be fish resources or marine mammals, birds, in that where they go in the ocean, it may matter to them that there isn't going to be a consistent response in the bottom of the food chain to temperature changes. For our study, we focused just on the temperature variability that we saw during the time series, but we are expecting that marine heat waves will become more common and perhaps more intense, and also that over time we expect a general increase in sea surface temperature. So understanding that the marine plankton communities are not going to necessarily respond the same way across the whole region is one important outcome of this study so far. I'm going to go downscale a little bit now and talk about the eddies in more detail, which is something that's always fascinated me since I first came out here. So most of the eddies that I'm going to be talking about are anticyclonic in the Northeast Pacific, and they show up on these sea surface height plots as positive anomalies. So this is a little inset from just off the northern tip of Hawaii, and you can see patterns of positive and negative sea surface height anomalies. So again, up to about 30 centimeters for the strongest ones above average. And the eddies I'm going to talk about, they're typically just a few tens of kilometers to maybe a couple of 100 kilometers in diameter. Some of them can persist for quite a long time, maybe more than a year. And because they're anticyclonic, they're circling, so they tend to retain the water properties within the eddy much more strongly. And we say that they decay gradually. So if you can imagine, you've got a little dome at the surface, and as they decay, they kind of shed from the surface and spread out gradually and sort of merge with the surrounding water. So again, they look static in these images, but they're actually a dynamic feature in the ocean. One advantage of looking at sea surface height from satellites is that this can be detected through clouds. Anyone who's spent any time in my part of the world, it's not that different from the UK, and there's a lot of clouds, so you don't actually get very good images of sea surface color, say, but you can get sea surface height, whatever the weather. So here's an image from 2007 showing an eddy sort of right in the center. That's the coast of Alaska off to the northwest there, that's Cook Inlet. And we can overlay our ship's route on top of these altimetry images and identify which of our samples were within or close to eddies. So we've got a group of samples there that went right through the middle of that eddy. So we can identify those and look at them in more detail. We've done that for the whole time series, so we have actually sampled hundreds of eddies in our time series. Now, some of these eddies we've sampled multiple times because they persist, and we may sample them subsequent months. But as you can see, we've got quite a range of eddies here that we've sampled to look at. So that makes this data set quite unique. This is one of my favorite images. It's just to the north of where I am. So this is the Haida Gwaii Queen Charlotte Islands, which is the northern part of British Columbia. And this is actually a color satellite image. And it's been colored to show the phytoplankton in the water. And what you can see, I think, in the center is this beautiful eddy that's been spun off of basically the topography of the land and the continental shelf just to the south of that big island there. It's spun this eddy off as the currents are moving up the coast. And what the eddy does is it takes this shelf water with it out into the open ocean. And you can see the swirling there of the patterns as the swirling of the eddy is mixing that ocean water with the shelf water. And shelf water typically has more nutrients in it because they come off the land in the rainfall. And that's why we see most of the fisheries production on the shelf, is because there's more nutrients in the water to fuel the phytoplankton growth and support the food chain. So what you have here basically is a parcel of coastal water that's been moving off the coast out into the open ocean. And just over to the right of that very pretty eddy in the middle, you can see an older eddy. It looks almost like a ghost of the other one, but that one is much older, so spun off earlier. But you can see it's still detectable as an increase in plankton-rich water for moving further out into the open ocean. So it's long been known that some marine predators can sense or detect these eddies as hotspots of productivity and can perhaps target them in their ocean migrations. So that's one thing that we wanted to look at. So we're in the middle of this part of the study right now, putting all of that data together from all of the plankton in the eddy samples. And what we've got here is the total diatoms and total dinoflagellates, so two groups of the phytoplankton that we record. This is a logarithmic scale, so there's orders of magnitude difference. But on the bottom axis here, we've got the inner core of the eddies, the outer core of the eddy. So, and then that dashed line there marks the edge of the eddy. And then the three bars to the right are increasing distances away from the eddy. And what you can see there is that the outer core of the eddy and just outside the eddy are the areas where we typically see the higher phytoplankton. Over on the right, the numbers go up again, and I suspect that that is mostly because of the influence of the shelf. So as you get further away from the eddy, at least on one side of it, you're actually getting closer to the coast. So that's one thing we have to look at and try and remove any effects of coastal samples. But you can see, I think, that it looks like the eddies have high numbers of phytoplankton, and especially on their outer rim where the eddy water is mixing with the oceanic water. And that makes them a very attractive place for predators to look. And if we move up the food chain and look at zooplankton, okay, this is a complicated graph. Each of these different colored bars represents a different group of zooplankton. And some of them do show a relationship with the eddy and some don't. But for example, the copepods, those little crustaceans which mostly feed on the phytoplankton, show a similar pattern, so that their numbers are at the highest in the outer core of the eddy or just outside the eddy, and they tend to diminish in the center and to the outside. So then if we move up to the Seabird data that we have, again, this is a big database of collected information over multiple years from multiple sources. And some of the seabirds do show clear associations with the eddies. So these are petrels, which will feed on krill and larger copepods, as well as fish and squid. And it's very clear there that their abundances are generally much higher within the eddies, and then they drop off as you go away from it. Now, full disclosure, not all seabirds show association with eddies. Here we've got Arctic terns, and their numbers are actually, they're avoiding the eddies, or it looks like they're avoiding the eddies. In fact, they're actually much more closely associated with coastal waters. So we have a database of many different seabird species, and looking into which of those show an association and which don't will be one of the things that we're going to be looking at. And one of the other things that we'll be exploring is what features of the eddies are important. So the origin of the eddy in time and space. So these eddies can be formed in various places along the coast. Does that matter? And when it's formed, does that matter too? If it's coming off at a time when there's not much nutrients in the water, they may not actually be hotspots. How big is the eddy? Is that important to marine predators if they're targeting them? And how old is the eddy? We can expect to see the strongest effects when the eddies are youngest, and as they decay in time, maybe a year after they've been formed, maybe they're really not any different, practically different from the ocean around them. And the strength of the eddy, how fast is it moving? How strong is it? Do those things make a difference? And obviously we'll be looking at the diets of seabirds and their use of the ocean. Is this part of their migration pathway? Is this close to their breeding colonies? To build up a picture of how this kind of mesoscale structure in the ocean might be important to these seabirds. So first of all, I want to thank everybody involved in the project, mostly my co-authors from the Farallon Institute and Earth and Space Research, and then also to the CPR analysts at the MBA for doing all the extra samples. I imagine that my name was mud a few times as they had to go into the archive and look for these samples that were collected within eddies and pull them out to be analysed. I really appreciate them doing that. Definitely to the ships for collecting the samples, because we couldn't do this kind of large-scale study without them. And thank you, everybody, for your attention. And I will take any questions if there are any.
Thank you, Sonia. Have you stopped sharing your screen? Has that come through?
I did. I tried to.
Yeah. Okay. No, that's great. Thanks for that. So yes, excellent talk. Some of that imagery is fantastic, and all the moving video worked as well. So that's
good. Excellent. It's always a bit nerve-wracking to put a moving video into a PowerPoint presentation.
Yeah. No, it all worked fine. So there's a couple of questions that have come through. First one was to do with the zooplankton community structure against the sea surface temperature. Has that only been applied in your area, or have we applied that elsewhere? I should possibly.
There's definitely been some. I mean, I think that the North Atlantic Survey has done different things. Sea surface temperature is a huge structuring factor for zooplankton communities. So certainly there's been a whole history of work from people at the MBA on looking at how plankton communities respond to temperature and global warming. So, there's a lot of those things that have been done. I guess we took a slightly different tactic, and we're looking at stability in community structure as opposed to changes in community structure. And that was our angle, was how stable are these patches of communities versus how are they changing. So,
okay. And my other question was you linked the sea surface temperature to the CPR tows. Clearly, with CPR tows, you don't have any control about when that is.
No.
Are there any sort of noticeable gaps in the sort of coverage?
Well, we made a strategic decision right at the start of the North Pacific Survey, because funding was somewhat limited. We decided we would focus on spring, summer and autumn and not do any winter sampling, partly because we know that at these kind of subarctic temperate latitudes, there's not as much plankton around in the winter. We also know that that's the time when the ships might struggle with samples, so you could spend more resources on trying to get samples at difficult times of the year. It gets really stormy in parts of the North Pacific in the wintertime. And some of our ships go into the Bering Sea, and there is some sea ice around in the wintertime. So that was another reason for avoiding winter a little bit. But it was mostly a strategic decision to focus the funding that we had on looking at the time of the year when there was more plankton around.
Yeah.
So yeah, so we are missing some winter information. That certainly is a gap. And then inevitably there's always, the odd time when the sampling doesn't work. But we've got pretty good coverage over those 20 years. So.
Excellent. Okay. Maya had a question about, can you tell us a little bit more about NASA's program, Life in the Moving Ocean, and how this project fits in with the aims of that program?
Well, NASA's actually been quite easy to work with. Now, I don't do that work. They don't actually. The way the project's set up is that most of the money is going to a US based organization, the Farallon Institute, who are working with us on this. But basically, they were very keen to pull in biologists, to use satellite data. Most of my colleagues are actual physical oceanographers, so they kind of roll their eyes when I start talking about biology. And if they ever see this video, they'll probably shoot me. So that's where traditionally a lot of satellite data has been used, and developing current models and things like that. So they were very keen to explore the way that biology can use satellite information and start to understand how, the dynamic, like I said, the dynamics of the ocean, how that might influence the biology that's living in it. So that was sort of the purpose of that program, was to to pull together. Most biologists we're looking at, we're looking at the organisms in the water, so to be able to pull that sort of satellite data together with that observations of marine animals.
Okay, that sounds a good program. Willie had a question about the fisheries data. Clearly, you're linking the two, and is there fisheries data associated with the eddies, and do commercial fishermen use oceanographic data like this to target certain species associated with eddies?
Yes, it's one of those kind of anecdotal things that if you talk to some fishermen, they know that that eddies are hotspots. One of the issues has been simply knowing when the eddies are there, and also how quickly a ship can get there. So there was a research cruise back in the early part of the 2000s that had been planned to actually go and sample the properties of eddies, but it's very difficult to do that. You can't predict exactly when they're going to be. Some years you have very few eddies, some years you have lots. So you're relying on satellite imagery, and then you're sort of organizing your cruise, but you've got to have good communication because the eddies are moving, and you can't see them when you're there. You can't see them from the surface. You've got to have the altimetry image to say, okay, you're right in the middle now we're sampling. But there was anecdotal evidence that yes, tuna, some other kind of attractive species, as well as the seabirds and marine mammals were targeting the eddies, which is one thing that's kind of prompted them as being hotspots. But whether fishermen are actually using that information, I don't know. Some of these are a long way offshore, and I suspect that they're still getting better catches, on shelf waters more than going out into the open ocean.
So they haven't been studying the results then, as far as you know.
I'm sure they have. I'm hesitant to say no. I'm sure there have been some studies looking at that.
Yeah. Okay, thank you. And Jennifer had a question. An increase in storms is being forecast as a result of climate change. Will this likely disrupt the formation of eddies or their longevity?
I am absolutely certain that it will affect their longevity. Whether and this again is not something that I can pull out references to right now, but there is a suggestion that more eddies are formed in warmer years. So while stormier weather may affect their longevity, you may actually find that there are more that are being spun off in the first place. So it might be it's one of those things I think we need to do more studying on to find out whether there are more of them, but they don't live as long. We might be able to get at some of that with the time series we have now, but we have to remember that satellite data is still relatively recent. We're talking really some of these things only 2-3 decades of data, and that doesn't encompass the whole range of variability in ocean climate that we're going to see. So we're still only going to be able to tentatively suggest these kind of patterns of relationships between climate and ocean conditions. But I'm certain that when it's stormy, these eddies will break down more quickly, yes.
Right, excellent. All right, thank you. I think that was all the questions we had. Oh, sorry, Maz has brought one in. Do the warm and cold periods you mentioned relate to El Nino years?
Yes, to a certain extent. So we do have this phenomenon in the is kind of periodic. So every 5 to 7 years, we might typically have a warm period in the Northeast Pacific, which is an El Nino, and then you have a cool period, La Nina, or that you have years that are kind of neutral. And so that is certainly enhancing some of the changes in warm and cold years. But it's not the only thing. The heat wave was exacerbated by an El Nino, but it started before an El Nino. So the heat wave started in 2014, and it wasn't part of that cycle. It was actually brought about really by some very stable winter weather, which didn't mix the heat that's stored in the deep ocean up to the surface because there weren't as many winter storms. You had a lot of heat that was still there come the next year, and then there was an El Nino in 2015 and 16, which just added to the heat wave by bringing in more warm water. So they can certainly exacerbate or exaggerate or mitigate the other ocean climate things that are going on. But yes, El Nino is a big part of that cycle too.
Right. Okay, thank you. And Tanya has asked about the future of the Pacific CPR survey. Are there any possibilities?
Great future.
Great future. Is there any possibility of extending survey south to California?
We did some, yeah. It's possible. I always have people who are interested in extending it, and for a while we were exploring the possibility of going down to Hawaii too, which would have been really attractive for a lot of people to do. So yes, there is talk about that. And it's always a possibility. We've been doing the same routes now for 15, 16 years, so there's perhaps a reluctance to change those routes, but we're always open for new ones to come in, for sure.
Yeah, brilliant. That'll be good. Right, I think we'll leave it there. Thank you ever so much for the talk. A brilliant talk. Various people saying
you're welcome.
Thank you for those. I'll just share my screen,
sure.
And then. Maybe if people want to say hello to you or anything, then
yeah, I can hang around. Yeah.
So I'll just yes, just to say that the next Marine Biology Live session will be with Sancha, who's a student at Plymouth University, and she's going to be sharing the student perspective on marine biology, and that's on Thursday, the 24th of September, back at 11 o'clock UK time. Unlike Sonia, we won't be dragging her out of bed to do the talk.
Much more civilized.
Yeah, just to mention again that these are ongoing talks of the MBA. The MBA is a membership organisation, and we'd be delighted if people were to join the MBA to take part in some of these sessions, but also for members to come forward and provide a Marine Biology Live talk themselves. There's space later in the year for that if people are interested. So I think we'll leave it there, and thank you again, Sonia, for a fantastic talk.
Really thank you.
A lot. Thank you.